Hello everyone, my name is Siqi Fang, and today I will be discussing the effectiveness of scaffolds and vascular endothelial growth factors in repairing and regenerating damaged tissue caused by conditions like bone disease. Each year, millions around the world are affected by bone-related conditions such as osteoporosis, osteoarthritis, bone cancer, and more. In the US, about 50 million people suffer from osteoporosis, and about 1.5 million individuals suffer fractures due to bone disease. So, why is this happening? Bone diseases, defects, and cancer reduce bone density, making them more liable to break. These conditions can be life changing to those who suffer it. First, their overall health may deteriorate as one fracture may not heal properly and lead to more in the future. Additionally, while the body is busy repairing the fractured bone mass, it may cause other injuries like cuts or bruises to heal slower. This could be an issue, especially if the patient is older, as it may further strain their immune system. Also, bone diseases are debilitating, particularly for the elderly, as it can prevent them from accomplishing simple everyday tasks independently. Lastly, paying for hospital bills, caretakers, etc., can be costly for those with a limited source of income. In this graphic here, you can see that in 2018, families spent 52 billion on osteoporosis related bone breaks. By 2040, it is predicted that 95 billion will be spent annually due to osteoporosis. If we can develop technology to alleviate the consequences of bone diseases, all this money can be utilized elsewhere. Here are more statistics. This one shows how these bone conditions may affect people aged 50 or above when their bodies are weaker and more susceptible to ailments. In this image here, we are looking at bone cancer survival rates within 5 years. Although 68.9% may seem like a higher chance of survival relative to other cancers, that is still 32 deaths out of 100 people. So, how do we combat these diseases? Let me introduce you to mesenchymal stem cells. Mesenchymal cells are non-specialized cells with great proliferation potential and capacity for self-renewal and differentiation. In other words, mesenchymal cells can be cloned and grown rapidly into other desired cells. As you can see here, these cells are first derived from bone marrow, then they go through self-renewal, also known as mitosis, and then they can transform into any of these cells down here. Here's another image that shows how mesenchymal stem cells can be utilized in tissue engineering. First of all, these cells can be derived from fat tissue or bone marrow. Then they are transformed into cells that can be utilized in different scenarios, such as local injection, where mesenchymal stem cells are injected directly into an area that needs tissue growth. They can be used to create tissue that can help replace defective bone mass or connective tissue, and they can be inserted into the bloodstream where they can flow to wherever the body may need them. Also, mesenchymal stem cells have the potential to undergo through osteogenesis, which is bone formation, adipogenesis, which is fat formation, and chondrogenesis, which is cartilage formation. This shows how versatile mesenchymal stem cells can be in regenerative medicine. Mesenchymal stem cells can also be derived from a variety of sources, making it less controversial than embryonic stem cells. For example, if taking these cells from the umbilical cord is too controversial, researchers can also obtain these cells from bone marrow donated by volunteers, which may be seen as more ethical. In this way, researchers can continue to improve medical technology involving stem cells safely and more efficiently. However, there are also challenges that need to be addressed, especially tumorigenesis. Tumorigenesis is the process of the development of a tumor which is caused by uncontrolled cell growth. As you can see, mesenchymal stem cells have the ability to divide and grow. However, when undergoing interphase, the preliminary phase of mitosis, where cells duplicate, uncontrolled cell growth may occur in the G1 phase if it is not regulated, or if the checkpoints are defective and don't signal the cell to stop growing. This could lead to the formation of tumors, which can become malignant and escalate into cancer. So, how do we minimize the risks of tumorigenesis? Well, we can use scaffolds as a guide to regulate and restrict the cells from growing in an uncontrollable manner. Scaffolds can act as barriers, keeping nearby tissue out of the treatment area while also keeping mesenchymal stem cell growth within the desired area. Scaffolds provide temporary support for the new tissue, giving cells an easier time to attach, grow, move, and change on these surfaces. As you can see in the image on the left, scaffolds can be used with cells and other biomolecules to create artificial tissues in vitro or in the lab. Then this artificial tissue can then be transplanted into the body. Additionally, scaffolds can be customized using 3D printing, allowing researchers to make flexible changes to scaffolds and maximize the efficiency of stem cells. As you can see in the image on the right, there are many approaches to scaffolds, and if one of these approaches don't work, there are also many others that researchers can experiment with. In this test, it is important to note that PLGA stands for the presence of scaffolds, and just looking at these results, we can see how the tests with scaffolding produced higher amounts of stem cell growth than the controls. However, it is also important to note the limitations of scaffold, such as its biocompatibility, as the raw materials could trigger the immune system, and technical challenges like vascularization required to maintain complex tissue structures. This brings us to another question. How can we reduce the limitations of scaffolding that are inhibiting the development of mesenchymal stem cells? Well, with the use of VEGFs, also known as vascular endothelial growth factors, researchers are able to solve the issue of vascularization that is present in scaffolding, allowing mesenchymal stem cells to attach more successfully to these structures. This improves both the efficiency and effectiveness of using mesenchymal stem cells to regenerate bone tissue, which can help mitigate long-term consequences of bone disease. So in this image here, different forms of VEGF were introduced into the chorioalitoic membrane, which acts like a placenta of an embryonic chicken. As we can see, these labels on the left are the different approaches to mesenchymal. These are the different approaches to VEGF implantation. And VN stands for no VEGF, while VS stands for the presence of VEGF. And just looking at the control here, we can tell that the tests with VEGF has noticeably more blood vessel formation compared to the ones without. Similarly, high densities of VEGF has more prominent blood vessels compared to low densities of VEGF. And this just goes to show how VEGF has a significant role in increasing the creation of blood vessels, also called angiogenesis. All right. While VEGF are beneficial to helping mesenchymal stem cells attach to target areas, they do have short half-lives of less than 30 minutes, requiring large amounts and multiple trials to utilize it successfully. However, the use of large quantities of VEGF should be avoided because it could lead to catastrophic vessel formation at non-target sites like dormant tumors. This could awaken the tumor and allow it to grow rapidly, which would escalate into cancer. This goes to show how VEGFs can be a double-edged sword, as their ability to create new blood vessels could revolutionize tissue engineering, but their ability to catalyze tumor growth can also be detrimental to patient health. Through the analysis of multiple different sources, it was found that mesenchymal stem cell growth that utilized scaffolds had higher success rates than their control groups, which did not include any external factors in the cell's growth. From most sources, it was noticed that tests utilizing VEGF along with scaffolding had significantly higher successes in bone regeneration than those without the biomolecule. As you can see in this image here, CMV is the test that utilizes both VEGFs and scaffolding. CM only has a scaffold, and the control did not have either of the two factors. And off the bat, just looking at these images here, you can tell how this test had more blood vessel formation than the other two. Also, looking at the bar graph, we can see that the test with the scaffolding and the VEGF has had the most number of complexes, the most number of junctions, the largest sizes, and the longest lengths of blood vessels. In this image here, this is an image of bone defect repair attempts in the hind limbs of mice. In test A, there was scaffold with VEGF. In test B, it was scaffold only. And in test C, it was a control, meaning it had neither. So just looking at this image, we can see how in image A, this bone has completely healed. There is no remnants of scaffolding around it. And yeah, it's just completely healed. However, compared to image B, we can see that over here, there's remnants of scaffolds that has not completely dissolved. There are also many pores in the bone and a few cracks in the bone, showing how this test may not have completely healed during this time limit. This also infers that test A is probably more efficient with due to the addition of VEGF. And in test C, we can see how this crack is still here. There has been no bone regeneration, um, showing how without any scaffolding or prevascularization, test C has failed to regenerate bone mass. This test is derived from the same experiment as the previous slide. This is just a closer up result. Um, and as we can see here, there's a lot of solid pink in test A, showing how there was a lot of bone mass that was regenerated. In test B, we do see some solid pink. However, there's also this transparent pink here, showing how the scaffold has not completely dissolved and that test B is not fully healed yet. In test C, we see a lot of purple dots, but not as much solid pink, showing that although test C did fail in the experiment's main objective, which was to generate bone mass, it did create lots of other connective tissue in its place. Despite these overwhelming positive results, it is also important to note that not all experiments conducted had shown these results. Additionally, a current challenge that needs to be addressed is the inability to translate in vitro or in lab experiments into in vivo clinical applications, as there are many factors that scientists may fail to account for in an artificially made lab setting. So, although these methods may seem slightly unstable now, many trials have shown promising results. Therefore, scientists should continue to experiment and work to develop safer methods of tissue engineering with mesenchymal stem cells, scaffolding, and VEGFs while still being aware of the unknown side effects and challenges that may arise. Thank you for listening to my presentation, and here are my references.